Well, man, thanks, Mike. I don't think I can top that. So you guys just want to go to lunch? We just start right here. Well, obviously, I'm honored more than you can possibly imagine, and surprised by this delightful award. And I thank everyone who showed support to me, because it means a huge amount to be included in this group of people who are so stellar, and especially including Janet Rowley herself. If you've never met Janet before, I'm sorry you couldn't. I want to tell one story. Jane wanted me to tell the story. So if we run over, blame Jane. I was visiting the University of Chicago. Janet had invited me to give some talks there. And so she was, like being the good host, would take me from person to person during the day. And I was noting some weird sound, and I thought, maybe I'm going nuts. I've worried about that for quite a while. And so throughout the day, we're walking around. There's this clunking, clacking, weird sound. I'm looking around everywhere, and finally I said, Janet, what's that sound I'm hearing? And she had this backpack on her back. And she goes, well, I'm going to go trekking in Nepal soon, and so I walk to work every day, and every day I pick a stone and put it in my back on coming and going. So she had this bag of rocks that she carried around all day getting ready for Nepal. So this shows how brilliant and practical she was. And I'm sure that if she was still alive today, she would be the first to sign up for the ICML trip to Annapurna. Oops, let's go this way. So when I think about Janet, there's a quote from the United States humorist Will Rogers that said, "It is great to be great, but it is greater to be human." And that summarizes Janet entirely. But I want to talk, digress a little bit about my feeling about this prize. I'm going to go here with a story that I'm sure has never been told in this conference through the years. And that is, I'm going to take you back almost 100 years entirely to Nome, Alaska, as a so-called race of mercy. So there happened to be a diphtheria outbreak in Nome. And at that time, that was a small outpost. I don't know if my pointer is working. Not. Oops, wow. Sorry. Nome is a small outpost in Alaska. And the diphtheria breaks. And there are 20 ampules of antitoxin in Anchorage. It's December. It's minus 30 degrees centigrade outside. There are no roads. Planes cannot navigate that. And though at that time the water around Nome would be frozen. So there's no way in at all. And so what they do is they quickly get a team of mushers. So there's 670 miles to go from Anchorage to Alaska. A team of 20 mushers with their dogs. And they quickly arrange to do the so-called race of mercy, where they're going to basically dog sled these 20 ampules of antitoxin to Nome. They did it in 6.5 days. At sometimes the weather got to minus 70 degrees centigrade. Scores of dogs died on the way there. But the last dog to make the trip was Balto. Now, Balto was what they referred to as a scrub dog. He really wasn't. He was just kind of one of the members of the team. 2 legs to go. For some reason, his owner decided to have him in the front of the pack. And so they went to the second to last leg. They got to the last leg earlier than anticipated, so they just kept going. So for the last 30 miles was there. They got the ampules there, all 20 of them, not a cracked ampule. And they basically rescued this epidemic of diphtheria that was killing children in Nome. Balto becomes famous. He actually has a statue there in Central Park in New York with the inscription, "Endurance, Fidelity, and Intelligence." Balto was taken on a tour around the United States as being this, wonder dog. Trouble is, everyone knew that actually the main work had been done by Togo. Togo was a famous dog. He had gone through the major the worst conditions for multiple legs, and everyone knew that the hero was Togo. Togo got no press. So I'm here accepting this award. I am Balto. Woof. So I want to talk some about CML and why it's still relevant. We know on the left there that's what we've done. Remarkable stuff looking at overall survival in CML over the time. But I think now that I'm doing more and more stuff with kind of CML adjacent diseases like AML, it's clear that CML really continues to be the model for a number of things. One is precision medicine. The second, I think, is understanding progression and resistance, and certainly in global medicine and how to democratize technology. So what I want to emphasize here is all the work we've done, it's still very relevant because the rest of the community is looking at us for sort of leadership on how to do things in these areas. And in discussing models, I want to both mathematical, biological, and the like. There's a quote by the famous statistician George Box that I think that's important to remember is, essentially all models are wrong, but some are useful. So I think that the big questions in CML are going from left to right. One is who responds to therapy. The second is who relapses on therapy. And the third is who can discontinue therapy. And some of those topics we've discussed already. Some we'll discuss throughout the whole sessions over the next couple of days. And when you look at diseases that are really looking at CML as a model, for the response to therapy, it's clear that AML is doing that. We'll talk about the AML precision medicine in the program in the NCI in a few minutes. Everyone wants to understand who relapses to therapy. That's really the issue of minimal residual disease. Huge issue in AML, ALL, CLL, and multiple myeloma. And who can discontinue therapy, that's really becoming important now. And if you go to CLL conferences, it's really one of the things they talk about, is CLL as being the new CML. And what can they learn from our experience to guide how they do trials and the like. So one of the things everyone is interested in now is so-called precision medicine. And when I started trying to do this stuff, I had this kind of model that I drew down in the middle there. Obviously, I'm no Sue Branford in artistic design. But the idea is you come in as a sick patient, you make data, and from the data, you miraculously pick a drug, and then you suddenly get this well patient, right? And when I started this, I thought it was this simple. It turns out to be enormously complex. Actually making the data is the easiest part. Finding the drugs, finding the cooperative pharma partners, devising the bioinformatics to link in all of the data, outcome and the like from various institutions to make this happen, enormously complex. But I think it's worthwhile doing, right? I think it's really, really important to try. And it reminds me of a quote here by the musician Sun Ra, the impossible attracts me because everything has been done and the world didn't change. Which is a pretty good quote for a guy who really thought he was born in Saturn. It's true. So our foray into resistance and progression started by looking when gene expression first came out. I apologize for those who are red-green colorblind, where we looked at blast crisis and compared it to chronic phase. And what you found is that there were about 3,000 genes that were differentially expressed with a p-value of p to the minus 12, so they're probably real. And so the idea there was that you could basically find genes that were predictive of progression and find the biological pathways, so you could both predict who's going to respond, who's not going to respond, and then do something about it, right? And this, I think, worked. What we have here is, again, showing those 3,000 genes. And on the right, we've got essentially a score of blastness, where you look at gene expression compared to chronic phase, and you can give every patient a score of blastness, blastitude, whatever. And so they're ranked there, and you can see the arrows on the far right, which I think this is still not going to work. No, sorry. Oops, wow, oh, it does that. Sorry. We're not going to try that anymore, folks. You think I'd warn. On the right are patients with those dots running up the side, are patients who the pathologist said were in chronic phase, who had therapy, went into remission, came out, now have BCR-ABL positive, but to the pathologist they say this person is in just chronic phase. And you can see that many of those signatures are not chronic phase, they're blast crisis. And this, I think, explains to why some of the early work by Simona and Sue and the like, who showed that people who relapsed before we had other second agents, relapsed very quickly, quicker than you'd expect. It's because they weren't chronic phase, that there's some shared expression pattern and biological pathways between progression and resistance that are driving things. Other groups have shown this. Mike's group, when he was in Portland, the Mannheim group, using CD34 selected CD0. Probably the most validating thing was that for us, because I always assumed that our data was probably wrong, was an elegant mouse model where they basically first induced BCR-ABL to get a chronic phase, and then had a sleeping beauty transponin that they released, which caused genetic bedlam everywhere. And if you looked in the mouse model about gene expression pathways and the like, that model pretty much overlapped these data. So that gave us an idea that that's probably something to it. Vivian did this work early on, showing that if you basically scored patients, you could kind of predict their outcome. Let's go to the left, the middle panel there. The blue dots are patients with newly diagnosed CML. So you'd expect chronic phase newly diagnosed CMLs to have a low blast score. The blast score is on the y-axis. And on the right are people who became resistant to imatinib, and they're about to go on an imatinib trial, right, to rescue them. But they look like chronic phase. And you can see none of those people look like chronic phase on their blast score. And if you believe that's true in treating with conventional therapy, it should probably also be true in transplantation. So retrospectively, we went to the right there on Hutch patients. Those are chronic phase CML patients, transplanted early. And if you give them a blast score, you find that those with a low blast score do great. People who look to be in chronic phase but aren't by the blast score relapse, right? So it tells you that there's probably something biologically to this. Sue Branford kind of stepped up the game, as she always does. This is looking at patients who are both an accelerated or blast phase and both don't respond well to therapy, and finds that the theme persists, that progression and relapse, at least, or progression and resistance, at least partway, are complementary. She found that the people who have more mutations at diagnosis do worse. Patients with new cryptic rearrangements and the like do worse. So again, it's the same theme of being able to look at patients early on and tell who might have more trouble and who might not. Many of us in this room participated in this study. This is looking at sort of the kinetics of response. And you can get all these various layers of response based on BCR-ABL disappearance over time. Dr. Stein here basically had like 5 different things. So again, with the idea of precision medicine, can you actually look at people when they walk in the door and tell what glide plane you're going to go on? Right, so there's a lot of ways to do that. In our lab, because all these things are expensive, right? I mean, all the sequencing stuff is really expensive. So what we've taken to doing in both AML and in CML is say, just take the outliers. Right, just study the outliers, because if you don't see a biological impact in the outliers, you'll never find it at all stuff in the middle, right? Because there's all these middle curves. They're very heterogeneous. That might be from clonal heterogeneity. It might be how people process drugs. There's a lot to that. So you just take the outliers and hope you find something. So this is a study I don't think I've shown here yet that we published in this year, looking at the ENS and D patients and taking those outliers. Basically, take people who had a fantastic response, MMR, which, by the way, was Tim's idea for a term. I remember the night he did that. We didn't think it would stick, but here it is. So take those people who have a fantastic response, MMR, and then the people who, like, that we've seen over and over again, like less than 3 months, they have very poor response. So you take the good and the out. So some things you find interesting is that if you look at the curve on the right there, the glide planes, once the 3-month mark hits, stay for 10 years, right? So there is very, very little crossover that once they start on their path. And if you look at predicting things, you can look at gene expression, you can look at clinical stuff. The AUC for the clinical ELN scores, et cetera, is pretty lousy. It's almost a flip of the coin about what curve you're on. But gene expression, if you just look at whole gene expression, 0.79 is pretty good for this kind of data, for an AUC. And if you add clinical stuff to gene expression, you don't do anything at all. So there's probably something to the pathways. If you then look at, well, what genes are up and down regulated in these groups, and if you do some mathematical modeling where you take the top genes and then you do, take one out, do repeat modeling 1,000 times and say, well, which ones score the most? So that's on the x-axis. You have to be one of these genes, you have to show up in at least 10% of the models. You find that basically all the top genes for good responders are all involved with the immune system. Right? The ones that are response and poor responders are all upregulated, are basically cell cycle genes. So I think that's rather remarkable, because it kind of suggests that at diagnosis, people who are going to be good responders have their immune system revved up, right? So that's pretty cool. You can then look at gene regulatory networks involved with response. And how you do this is you take transcription factors and you focus on the transcription factors that are either up or down regulated. And then you call it regulon the genes that are sort of downstream of that. And so you kind of contract those going up or down. And then you can kind of build a model where you take all patient samples, and then those marching down those trees, you can see whether these regulons are up or down regulated, and does that force them into a pathway. And then you can predict, who's going to respond, who's not going to respond, and then compare it to the actual observation. And when you do that, you get, on the right box there, predictor positives and negatives are quite good. And if you look at the odds ratio, which is looking at the outliers there in the 2x2 table, the odds ratio of a good or bad response are 10-fold, which is pretty darn good. If you look at the genes that are upregulated in, again, the good versus bad response, they're all immune interferon genes involved in immune pathways, or targets where we have drugs to, IDH1, the Burton tyrase kinases, and P53s. So there is something that we may be able to look at patients from the get-go, and with sensitive techniques, determine who's going to do good or bad, and go from there. Now, one of the ways that is interesting to look at response, look at resistance, is a better way to do gene sequencing. So as you know, with most next-generation sequencing, you're sort of limited in sensitivity by the error rate when you make the libraries. So if you make regular libraries, the error rate is roughly about 1 in 1,000, which is why you really can't get reliable data with VAFs less than that, right? Really 1 in 100 in most cases. And so it's especially bad with cDNA. So you take mRNA, have to make cDNA, that puts in a ton of mutations. We estimated that in one study that for every 24 molecules you get an error, right? So you can really kind of have a jackpot mutation and get a false positive. So this is work that Mike Schmidt did when he was at the University of Washington. They've now spun off to a company. This is so-called duplex sequencing. Coleman Lindsley, who will be talking later on today, go see his talk because he's terrific, has his own variation of this that's quite elegant. And basically what it does is you build libraries off of both strands, and the only thing you call a mutation is within the complementary mutation in the exact same spot. So your error rate goes from 10 to the minus 3 times 10 to the minus 3 times the complementary. So you get, theoretical error rate is 1 in 10 to the 7th. The scattergram there shows a patient with a 359 mutation. So you've sequenced through the whole Abl kinase domain by conventional sequencing. That's the top one. And you can see for every location, you get a call of a mutation if you look down low enough. The bottom is duplex sequencing, where you only see the needle in the haystack. So exquisitely more sensitive. Mike's gone on to show a lot of interesting things with this. The early work was done before single-cell sequencing, looking at the different types of clonal heterogeneity you can get. And it basically is every pattern where you can get so-called chip mutations, and then you get BCR-ABL onto that to activate the disease. One case on the top is a CML clone that gets another mutation like AXL1 and the like, and then you can have the thing on in the middle where you can have a pH negative clone with other mutations, AXL, GATA, etc., coexisting with BCR-ABL rising and falling. So you can really start to get into sort of exquisite sensitivity of building these clonal structures. On the bottom is looking at sequential sequence mutations in people with multiply treated on the ponatinib trial. And again, you get the same thing. You basically can get down to really 1 in the 4th to 1 in the 6th, and really be able to track these mutations, very, very carefully. It also shows to why sometimes when we do conventional next-generation sequencing, we get mutations that don't make any sense on the context of therapy. You say, why are these mutations there? And they come and they go. They don't make any biological reason. It's because they're likely artifacts from the sequencing technique. So you don't have to reinvent Darwinian selection. It's all technology. This has now gone on to pave the way for studies of next-generation sequencing in other diseases. On the top is looking at a patient with CLL treated with, the colors are multiple BTKs that I cannot pronounce any of them. So thank you for that, pharmaceutical companies. But it shows that for any one patient, there are multiple mutations in all of the resistance genes. Now this is by bulk sequencing, so you don't get the flavor of the single cell work. Kathy, who was at Harvard, has done some beautiful work on that. But this gives you exquisite sensitivity on looking at the multiple genes there are at a very early stage. On the bottom is work at the holy grail that we work with, is MRD. We're comparing duplex sequencing to the standard that we do in AML, which is flow cytometry. And what it shows in most flow cytometry is that the relative risk of relapse for flow cytometry positive is around 2.5. With duplex sequencing, that relative risk jumps to 5 to 7. And so you can see there the green curve, the dotted line, are people who are flow positive. The red curve are the patients who are flow negative, but next generation sequence positive. So much more sensitive in defining who's going to relapse and who's not going to relapse. This has led, in chasing the CML experience, to two projects that I'm involved with. I've grabbed Coleman Winsley and Chris Horgan to be kind of the brains of the operations. And it just reflects how much the NCI in the US is interested in using CML as a model. The first is the AML Precision Medicine Program. That in that structure, every AML that walks into one of the cooperative groups in the United States will have rapid next-generation sequencing and cytogenetics. Those will both be done within 3 days. And they will go on specific trials based on targeted therapy. So the idea, again, like CML, you're going to target their lesion. And for these trials, they're not going to be these giant phase 3 trials. They're all going to be phase 2 trials based on MRD as an endpoint. So like we did in CML, we're going to try to turn, where we turn 10-year trials into 1-year trials, here you're trying to do the same. Is can you get enough of a signal early on to really pave the way for drug development? The second is the foundation of the NIH is sponsoring a giant program in minimal residual disease in AML, and that is really trying to make these hard, sensitive assays, much like we did in BCR-ABL, mostly Sue's work with the international scale. And so that's the model that they're pushing for, and we have lots of pharma and biotech backup on this, is can we get a robust marker that can really drive how we measure outcomes? So another area, I think we're still on time here, that we're looking at is better, newer ways to get more information out of the genome. Because right now we know that in CML and AML, cytogenetics is very important. The question is, can you get more data out of conventional cytogenetics? And so this is a method called proximity ligation. What it amounts to is you take cells and you formula and fix them. And then you circle ligate them and then sequence. And what you'll do is if you just have no translocation, you'll just get your own sequence of that particular stretch of chromosome. If you have a translocation hidden there, you'll get part of one chromosome and part of the other chromosome. And so this just kind of shows why you want to do it. The sensitivity of karyotyping obviously is pretty lousy. You're really restricted to 10 to the 7th base pair chunks of DNA to be moved around to see it. Fish has more sensitivity, but you're stuck with what you're looking for, right? If you don't have a fish report, you're not going to find anything new. This technique really can get down to about 1,000 base pairs of resolution. So the question is, and this is kind of how you look at it, if you imagine these are chromosomes X and Y, in unity, how the data dots, each dot is a sequence here, should kind of fall on unity, right? So you shouldn't see any scatter from if you're just sequencing normal. But once you get these things that move off from unity, those are unique translocations. For instance, you've got one here with a chromosome 2 mixed with a chromosome 5. And so then you can go into that and harvest those dots and kind of get what the sequence is. And this is just an example of how that works. Unless you did this daily, you would not be able to tell what those are. You would just know that they're not normal, but you have the sequence information, you can see what they're at. We first started doing this as work we've done with Vivian on AML and finding that you have, in fact, you can catch things like the regular breakpoints on the left. The one on the right is an inversion of ABLE. The one on the far right is a very, very big gap in ABLE. So you can start getting variations of this. But very quickly we realized that, well, the problem with CML is that it's so genetically pretty silent that it's going to be hard to tell what's false positive and what's false negative, right? And so we moved on to AML, where we took 50 samples that have had FISH done, cytogenetics done, CGAT done, and the like, and then blindly did this on. And we've shown that about 12% you actually recategorize. Most of them are normal cytogenetics where you find new lesions. Many of those are structural lesions. Few of them are recurrent. So the question is, are those real? How can you use them? And the idea is that we're trying to do something called a chaos score, where basically you can sum up the number of mutations around and then kind of use AI to kind of build a score. So you could then imagine taking intermediate risk CML and dividing that into people who have lots of mutations or on a cytogenetic level, and those that really are clean. And maybe that's why intermediate risk AML is a 40% survival. Maybe you can break those up into 100% and then 0%, right? So we have money to do 500 cases. We've got about 100 done. We'll be doing AML, we'll be doing chronic phase CML and blast crisis. And we can starting to compare things like AML versus myeloid blast crisis. Lastly, I'm going to go through this pretty quickly because Mike hit it. One of the major things we've done, obviously, is the work with global. And so this was originally work we started 20 years ago with the Max Foundation. Max Foundation made an agreement with Novartis that if they could diagnose a patient with CML in an area that didn't get tyrosine kinases, Novartis would give that person this free drug for life. And I think they did it because they figured that Pat would never be able to pull it off, right? We've got involved, as others have got involved. On the right shows how powerful this is. This is 60,000 people on the GPAP program. And what we've done is looked at survival based on like when they first came into the program to when their last time they picked up a TKI, assuming that they're alive when they do that. And what you find is that the survival in these low and middle income countries who are in this program is essentially identical that if a CML patient was treated in Seattle, which to me is just absolutely mind-boggling. Mike alluded to this before. We started off by developing the Cepheid BCR-ABL assay in our lab. I don't have any commercial involvement with them. That obviously was huge because the WHO rolled out Cepheid machines, so that increased the ease of testing dramatically. CML is, again, the forefront. Assays are being developed now with Cepheid cartridges in breast cancer, other myeloid malignancies, with the same idea of pushing these out into areas of need. Mike alluded to this on the right. This was the study we did showing that spots work. This was done with Sue, where she would do a test on fresh blood. We put it on paper, send it around the world. Sometimes it took up to 4 months to get to Seattle by snail mail, but the correlation is quite high. We have now done this on over 2,000 patients from 60 countries, where they're diagnosed, they get free drug for life. We figured out how to do this on all the mutations, both in ABLE and all the myeloid panel mutations. So we can so people who want to have myeloid genes checked, translocations, we can do that all in blood spots. And it really is, now when we are working with developing countries, we may not have to buy them refrigerators, freezers, and the like. We can really do studies with shoeboxes because these things will last, for forever. I'm going to go through this quickly. This is what we call the medical leech, which Erwin Bethier there from a company called Tassa, that's my office. If you want to come visit, I'll give you an espresso too. It's a device that you basically can put on your arm, and it collects blood really painlessly. And so on the right here is a study that Vivian did, taking patients who have the BCR-ABL tested by conventional means, and then having them do the BCR-ABL on their arm. It draws blood into a dried blood spot, and it looks really, really good. We eventually did this for thinking of low and middle income countries, but really, given the pandemic and the like, this is something that you can use everywhere, because you can prevent people from having to come into the clinic. You can, theoretically, you could call up your drone, call up, Amazon, deliver it by drone, deliver it back to the lab. So my last few slides, hopefully this will be worth it. We're talking about democratizing technologies to the world. And so this is our work with something called a nanopore, which is this really nice little slick box that's actually a sequencer. It's about that big. I usually bring one to talks, but they look like a bomb, frankly. And so I thought it would be very difficult to talk to a customs agent or something and try to explain what nanopore sequencing was, and not miss this talk being in jail. But what it is they have constructed, this Oxford company, pores that are basically biological pores that they proprietary have altered, right? So thousands of pores. When you turn on the juice, since DNA is polarized, right, it runs through the channel. And it runs fast. You can do 250 base pairs a second. And you don't need to amplify. It'll just drag it through. And so it's really rapid, it's really fast. It makes errors on a point mutation basis that we've gotten down to about 5% now. But for translocations, it's great, right? Because you're going to see something that's not there. The problem is, as you might imagine, even if you had 100% BCR-ABL bearing cell, most of the genome that you're going to be cranking through these pores has nothing to do with BCR-ABL, right? So most of your sequencing is for naught. But you can do clever things. So on the right is basically a technique where you put in guides and use CRISPR technology. So you cut out the chunks that you're interested in using probes that you know. We've gone up to, I think, 12 different targets now. And to that, you also add a probe that's a protein that actually makes it a little bit more likely to grab the pore. And so you can enrich by about 1,000-fold by doing that. And what happens is this thing goes through, as it goes through the pore, since it's an electrical field, depending on what base it is, you get a squiggle in the electrical field. And that's the call of what base pair it is, which is really, really pretty clever. And if it jams up, it changes the electrical field and kicks it out. So it's really, really pretty cool. This is just an example of, published recently, of an assay we have where you can do APL 821s or CML in real time. And you're seeing that most of the reads are happening there really in less than a couple hours, right? So this is something that's really nifty because it's transportable. You just plug it into your computer to do. And so we think that that's really a way that you can get some of this technology out there. This can be used for BCR-ABL, again, leading the way. Two applications here. One is Tim's group and the like have shown that using DNA-based MRD may have some advantage. It's a pain in the butt to do because the potential translocation is so large, you have to do multiple primers and the like. This is a way that you can actually grind through and get the translocation really fast. So for MRD, it's great. It's also great in, we're working with Kathy Smith at UC San Francisco doing BCR-ABL stuff and single cell work. The problem there is that you can find low levels of mRNA sequence in all these various cell populations. The question is it real or not? Just because you can do it in two separate experiments, it doesn't mean anything, right? Because as Mike showed with the fish, the whole point of that was that in most of these multidimensional things, the reproducible noise is much stronger than the biological signal. And so you have to have some other things. So what we've done here is you can take patients and quickly construct DNA-based primers to them. And then when you do the single cell, you can have a control that you're picking up mRNA signal in the same cells that have a DNA signal. So my last point. This is great, but areas of the world still are struggling because not everyone has internet access. On the left is a map that Cecilia Young made of internet access in February. You see the big areas in low and middle income countries. On the other hand, almost everyone has a cell phone. So one day we were having one of our meetings with our computer people, and we were saying how great it was, and boy, someday, it'll be amazing. You'll be able to do this on a phone. And there was dead silence after the laughter stopped. And one of the people getting his PhD thesis says, I think I know how to do that. So here is, as far as I know, the first time ever that's been shown is sequencing on a phone. On the left, you've got a nanopore unit that's running. It does the sequencing, and it shoves the sequencing information by Wi-Fi into the phone. The phone is doing all the alignment. The phone is doing all the base calling. And on the box there is the first sequence, BCR-ABL being detected in the sample at about 2 hours. So this is a way that you could actually think about actually really, really democratize sequencing, is you could do it and just have it sent to your phone. Right? And so what my task is, Mike's giving you $100 for if you can identify that picture. I'll do similarly. We want to know what the best ringtone is for identifying that your sequencing is up. My last slide, and this is where I thank everyone. Because I couldn't possibly put everyone's names down, because I would be in mortal fear that I would forget someone. So this is just a reminder of CML as a model of what we can do as a community. So on the left is, when I started doing this is a 1 month supply of transplant medications post-transplant, right? I started with Don Thomas, right, one of the gurus of transplantation. Don's favorite disease was CML. I inherited his experimental pathology lab. So that's what really got me started in this. But what we have done, not me, you have done, is turn that bag of pills into this one pill on the right there. Albeit a giant pill, it's about the size of Tim's head, 5 of Tim's heads, which is, I guess, no surprise that adherence is a problem in some patients. But I thank everyone for allowing me to contribute to this field. But I think the thanks really has to be turned around, and on behalf of all the patients, thanking you all. It's a quote by Margaret Mead on the bottom, famous anthropologist, that I think really summarizes things. Never doubt that a small group of thoughtful, committed citizens can change the world. Indeed, it's the only way that ever has. Thank you.
I think we can have a few questions, maybe 1 or 2, until I get a stern look from Jorge.
People are hungry.
Come on, guys, at least one question.
Yeah.
What's your favorite gadget at the moment?
A tennis racket. Actually, my new knee is my favorite gadget. We do this stuff, right? So I think that we do it because it's fun, right? My dad was sort of an inventor. I think what the field is what hopefully one of the reasons I don't retire. My wife always asks me why you're not retiring, and it's basically there's all sorts of questions I wanted to ask for years, right? Single cell stuff. We presented a paper here, a talk in 2011 on single cell, right? When it was just brute force. But the whole idea is that we're developing fun things where we can actually ask these questions that I had no way of actually getting to the answer of, right? But technology allows it. So I think where the field is going is eventually, if I had to predict, is that everything's driven by Darwinian selection, right? And so what we have to think about is not looking just at the cancer cell or not looking at it's an ecosystem. And what we have to do is figure out a way to manipulate the ecosystem in our patient's favor. Because right now, with Darwinian selection, Darwinian selection is just working for relapse. It's amazing that everyone doesn't relapse, right? You're basically putting a selective pressure. Of course, some people are going to get out of the way. So the idea is how to turn natural selection in our favor instead of the cancer's favor. I don't know how to do that. But I think we're getting now some of the tools where we can really start to really look at the landscape of the ecosystem and how it changes at time.
I think this was a fantastic closing statement, so thank you, Jerry, again.
Thank you.
All right, we go for lunch now. We're running behind, so, but remember we have a